ATDC was initially described in the hunt for the gene responsible for the genetic disorder ataxia-telangiectasia. Cell lines from AT patients demonstrated that hypersensitivity to irradiation in ATDC was initially isolated based on its ability to confer radiation resistance to AT fibroblasts. ATDC is located on chromosome 11q23 and has been shown to be overexpressed in bladder, lung, ovarian, as well as endometrial cancers. There were no previous studies that were performed to determine the role of ATDC in any normal physiologic processes or alternative to other cancers. Our lab has recently discovered that ATDC has an important role in the oncogenesis of pancreatic cancer by mediating the Wnt signaling pathway. And investigative efforts in our lab are also exploring the potential role of ATDC in the DNA damage response. To study the role of ATDC in pancreatic cancer stem cells specifically, we have employed in vitro sphere assays for our experiments. It has been shown that the ability of cells to form spherical colonies called tumor spheres in non-adherent culture conditions is reflective of cells with self-renewal capacity. We have found that these triple positive CD44, 24, ESA positive cells form such spheres that we determine tumor spheres, while marker negative cells do not. These triple positive tumor spheres can be passed multiple times without loss of sphere forming capability, and so demonstrating self-renewal capacity in vitro. Immunofluorescent imaging of tumor spheres that are that are formed by these triple positive cells demonstrate that the ATDC protein is highly expressed in pancreatic cancer stem cell. And using a lentiviral-mediated silencing RNA delivery, we were able to suppress ATDC expression effectively in these triple positive populations. Following the suppression of ATDC expression in pancreatic cancer stem cells by silencing RNA delivery, the sphere-forming ability of CD44, 24 ESA positive cells was consequently suppressed upon serial passaging of these cells. This data suggests that ATDC plays a significant role in the self-renewal capability of pancreatic cancer stem cells. We are currently exploring if this is mediated by the beta-catenin pathway. In this next experiment, we examined the ability of gemcitabine and/or radiation to induce apoptosis in either control or ATDC silenced cells. Viable cells illuminate green, and the apoptotic cells are orange. In the top panels, a limited amount of apoptosis is induced in tumor spheres from these triple positive cell lines, following a single therapy of gemcitabine and radiation. An additive effect is demonstrated when combinatorial therapy of gemcitabine and radiation is used. In the bottom panels, however, the same conditions were applied to spheres that silenced ATDC expression. As you can see, silencing ATDC enhances the effect of gemcitabine alone, irradiation alone, and the combination therapy of gemcitabine and irradiation, compared to those spheres with normal expression of ATDC. The quantitative measurements on cell viability of individual cells within tumor spheres showed an approximate 50 to 75% increased cell death following exposure to these DNA-damaging agents when ATDC was silenced. So to understand the specific mechanisms by which ATDC sort of abrogates this DNA damage response, we look to see if ATDC played a role in the DNA damage signaling cascade. We know that specific DNA damaging signaling proteins including both ATF and gamma H2AX, traffic to discrete nuclear foci of DNA damage. In this experiment, we examined if ionizing radiation caused ATDC to also traffic to these DNA damaged foci. First, looking at the figure on the left, the top two rows represent untreated cells, and the bottom two represent cells treated with 5-gray radiation for 1 hour. ATDC expression, either wild type as noted as ATDC plus or silence as noted as ATDC negative, is depicted on the y-axis. In response to radiation treatment, ATDC clearly traffics from the cytoplasm into the nucleus to discrete nuclear foci and co-localizes with ATR. Silencing ATDC did not alter the ability of ATR to traffic to these nuclear foci, suggesting that ATDC may work in parallel or downstream to ATR. Similar results were observed with gamma H2AX. We observed ATDC trafficking from the cytoplasm into the nucleus following radiation treatment, phosphorylation of gamma H2AX, and the co-localization of ATDC with gamma H2AX. Silencing ATDC does not appear to alter the formation of these gamma H2AX foci, suggesting that ATDC also functions downstream or in parallel with this signaling protein. So in conclusion, we have demonstrated that pancreatic cancer stem cells are resistant to radiation and chemotherapy with subsequent enhanced tumorigenicity when there is enrichment of cancer stem cells following radiation and chemotherapy. We find that ATDC is highly expressed in pancreatic cancer stem cells and confers relative resistance to radiation and chemotherapy. And based on our data, we believe that targeting ATDC has significant therapeutic potential in the treatment of pancreatic cancer. Thank you very much.
Thank you very much, Dr. Lee. I'm afraid you've used up your question time. We're running quite a long time. So if anyone has a burning question, if they could ask Dr. Lee later.